So, good morning. Also, thank you for the invitation to come here. It's really a wonderful place. It's really a pleasure to be here, in particular to hear all these first-person accounts of interesting and important science in our field. I think it's kind of a pity these talks haven't been recorded, because they'll form a good record of what went on. It's a real. They are being recorded. Uh-oh, I better be careful. Okay. Well, good. Will they be openly available to everybody? Yeah. Okay. Okay. Great. Fantastic. Yeah. Now I'm nervous. Yeah. Okay. The task we were given was to give you a historical overview or introduction to the field. My field is a little younger than most of what you've heard, but I'll still go back to the beginnings of that field. And then after giving you this really long, largely historical introduction, I will then talk about, so after I've looked in the past, I'll then talk about some current and future directions that the lab is taking. So I'll give you a broad overview of where all this is. So the TOR field is has been a very rapidly growing field. As I said, it's much more recent than the other famous signaling problems in this field, like G proteins. The TOR is much younger than G proteins, and this rapid growth of the field is actually the point of this first slide. Now, this over here is the very first ever model of TOR signaling published. It's one we published shortly after our discovery of TOR in 1991. I'm actually embarrassed I published this model. It reminds me more of a Gary Larson cartoon than an actual scientific model. It says TOR exists, but question marks above, question marks below. We really had no idea what it did, other than being the target of rapamycin. Today, this story is considerably more complex, and this is already hopefully out of date. I've stopped drawing models. It's gotten so complex. But what I'd like, and this increase in complexity has been driven by this exponential increase in publications in this area. And this, in turn, has been driven by the fact that TOR turned out to be both fundamentally important and clinically important, certainly much more so than we suspected when we first stumbled across it back in 1991. So in the introduction, what I'd like to do is take you back to 1991 and gradually work you through the years until you have an appreciation of which resembles what's over here on your right. And I'll layer on the complexity as we went through it historically. So the story actually started before 1991. It started in 1965. And this is when a group of scientists set out from Montreal to Easter Island, also known as Rapa Nui, to collect soil samples. And what they were doing is they were prospecting for exotic microbes which would produce novel metabolites, secondary metabolites, which they could then develop into a drug. And in this case, what they're looking for were antifungals, novel antifungals. And they did, in fact, isolate such a compound from a soil bacterium, which they called rapamycin, after Rapa Nui. But while they're developing as an antifungal, they learned that it had immunosuppressive properties as a side effect. And of course, suppressing the immune system is the last thing you want to do to someone with a fungal infection, so it was quickly rejected and almost forgotten. Until some years later, it was then rediscovered by two transplantation surgeons, Roy Collin in England and Tom Starzl in the United States, who now repurposed it for the very reason for which it had been originally rejected. So they developed transplantation surgery as a viable medical procedure by introducing immunosuppression into the clinic. And rapamycin was one of these immunosuppressive drugs, along with cyclosporin A and FK506, which revolutionized transplantation surgery. So rapamycin, although it was originally isolated as an antifungal, it first became famous as an immunosuppressive drug. If you go to Rapa Nui, you'll actually find a plaque which commemorates picking up this handful of dirt in Easter Island. It's in Portuguese, and it says, "In this place was obtained in January of '65 a sample that not only allowed the isolation of rapamycin, but also ushered in a new era in transplantation surgery. So this, of course, marks the fact that rapamycin was developed into an immunosuppressive drug. I like this plaque because, to me, it illustrates the mysterious ways in which science advances. I don't think anybody would have predicted in '65 that rapamycin would have led to what it has led. So rapamycin today has applications in 3 major therapeutic areas. As I told you, it first became quite famous as an immunosuppressive drug, but now its fame in immunosuppression has been eclipsed by its use as an anti-cancer drug. And now it's also used for cardiovascular disease, treatment of restenosis after angioplasty in particular. And this is, by the way, what rapamycin looks like, this natural compound secreted by a soil bacterium. I can also give you a timeline of these clinical applications of rapamycin. And I like to show this to give you a feel for how recent much of this is. Rapamycin was first introduced as an immunosuppressive in 1991. It was then used for treatment of restenosis about 15 years ago, and about 10 years ago it entered the clinic as an anti-cancer drug, first in the treatment of renal cell carcinoma, and this is the product, Toricel, produced by Pfizer, and now Novartis has its version on the market called mTOR, which is not only for renal cell carcinoma, but now a number of other kinds of cancers. So how did we discover TOR? TOR, of course, stands for target of rapamycin. It is the protein which rapamycin binds, and it is this binding which is responsible for its immunosuppressive action and for its anti-cancer action and for its anti-restenosis action. So how did we discover TOR? Well, this all started with an outstanding postdoc who joined the lab in the late 1980s by the name of Joe Heitmann. He was an MD, PhD. He was interested in these drugs. They were creating a lot of excitement in those days because immunosuppressive therapy just became a reality, but nobody knew how these drugs worked. They knew they could be used in the clinic, but nobody knew what the molecular mechanism of action of these drugs were. And he was curious to find out how these drugs actually worked once inside a cell. In Basel, we were fortunate to have another colleague who worked at Sandoz, and one of their major areas of research was immunosuppression. They're the company who had cyclosporin, the other approved immunosuppressive on the market, and they're, of course, interested in understanding how immunosuppressive drugs worked as well. It was very difficult to obtain rapamycin in those days. It was not commercially available, so we were lucky to have Sandoz and our colleague Rao in particular in Basel, who could then give us some rapamycin. So we could start working on this in the very, very early days of rapamycin. So what Joe and Rao then did was something extremely unusual. As I said, it was people were trying to understand how this drug worked. It was a very competitive area, driven by the fact that this was a new immunosuppressive drug. And what everybody else was doing was they were working with mammalian cells to understand how this drug worked. And of course, it's perfectly understandable. After all, the drugs were being given to humans. But what Joe and Rao did, which was extremely unusual and I think risky, was to say, we're not going to work with mammalian cells, we'll work with yeast cells, making the assumption that whatever the mechanism of action of the drug is highly conserved all the way from yeast to mammals. And if we work with yeast, we can use yeast genetics to crack this problem. In those days, you could not do the genetics with mammalian cells you can today. So that was their risk that the mechanism was conserved. So, and of course, this was somewhat mitigated by the fact that something many of our competitors had forgotten, conveniently forgotten it seems, was that rapamycin had originally been isolated as an antifungal, so it acted on cells like yeast cells. And of course, Joe and Rao knew this. Nevertheless, our colleagues thought this was completely physiologically irrelevant to give this human drug to a yeast cell. Why would you ever do this? This just gives you a sense of how this was viewed in those days. Today, it's not so uncommon to see simple model organisms in biomedical research, but in those days that was not so common. So Joe isolated some spontaneous rapamycin-resistant mutants, a very trivial experiment, simply made a plate with rapamycin in it. They first saw that rapamycin affected growth of our lab yeast strain, and then selected the mutants which grew in the presence of rapamycin. Very, very trivial experiment. And what he obtained were mutants defective in any one of three different genes, FPR1, TOR1, and TOR2. And this has a different name from these two because this is a gene that Joe had already characterized in the lab. It encodes FKBP, the FKBP binding protein. He'd been working also on the FK506 immunosuppressive drug in the lab. And FKBP was the drug that FK506, was the protein FKBP. FKBP was the protein that FK506 bound. So this is why this gene had a separate name, was a gene already known to us, but then he identified genetically two novel genes, which he called TOR1, TOR2, for target of rapamycin. What was unusual here is that these mutations were recessive and extremely common. These mutations were dominant and extremely rare. So why do we have mutations in three different genes, some very common and recessive, some very rare and dominant? Well, the answer to this question relates to the mechanism of action of the drug, and that's shown here. So this is again rapamycin, now drawn in a space-filling configuration. It's a very lipophilic molecule, so it simply diffuses across the plasma membrane. And inside the cell, it binds this very small 12 kilodalton protein called FKBP, the product of the FPR1 gene. And this protein is absolutely essential for drug action, but not for cell viability. So the toxic agent in the cell is not drug alone, it's drug in complex with FKBP. Drug alone does nothing. So since this protein is absolutely essential for drug action, but not for cell viability, any simple loss of function mutation in the gene encoding this protein gives complete rapamycin resistance. And that's why mutations in the FPR1 gene encoding this protein were extremely common and recessive. TOR, on the other hand, and this is just a very small part of TOR. TOR is 300 kilodaltons in size, and this is 12 kilodaltons. So you can see if we had the whole thing drawn, it would probably fill a good part of this stage. This TOR, unlike FKBP, is absolutely essential for viability and can therefore tolerate very little mutation. And every mutation we identified fell in the exact same codon, which specified a key residue in this alpha helix, which was a contact site between rapamycin and TOR. And the effect of these mutations was to prevent the binding of FKBP rapamycin to TOR without otherwise affecting TOR activity. And this is then why these mutations were so rare, because they're confined to a single codon. And of course, they're dominant because these alleles conferred rapamycin resistance even in the presence of a wild-type copy of the TOR. So these mutations were very informative. They not only contributed to elucidating the mechanism of action of rapamycin, the fact it forms a complex of FKBP, it also led to the identification of the rapamycin binding site in the TOR protein. And finally, and most importantly, it led to the identification of TOR itself. And that came from when we, using the mutations as marks in the genome, we were able to clone the TOR gene. And we cloned and sequenced the TOR genes from yeast. The first thing we found was that the two TOR genes are highly homologous, 70% identical, and they turned out to be the founding members of this novel class of atypical protein kinases called PICs, for PI kinase-related protein kinases. And the reason they're called that is that they all resemble known lipid kinases, PI3 and PI4 kinases in particular, but none of these has ever been shown to have lipid kinase activity, and all are in fact serine threonine protein kinases. And to this day, we don't know the significance of this resemblance to lipid kinases. Since this early discovery of TOR in yeast, it's now been found in all eukaryotes, all the way from yeast to humans, including worms, flies, and plants. And the name TOR has been kept throughout based on the yeast precedent, but now there's always a prefix to indicate what organism the TOR is from. mTOR means, or maybe I should say used to mean mammalian TOR. Now this, as of a couple years ago, the genome database people have mixed things up by calling it mechanistic TOR to standardize nomenclatures across all organisms. So mTOR can stand for mammalian TOR or mechanistic TOR. This is something that got me very upset when they changed the name, because there are thousands of papers which indicate mTOR as mammalian TOR, and now there are papers where they refer to yeast TOR as mTOR, so it's getting rather confusing. In any event, yeast also turned out to be rather unusual in that this simple eukaryote actually has 2 TOR genes. It's the only organism with 2 TOR genes. All others have a single TOR gene. So for once, the simple eukaryote is more complex. This complexity actually helped our analysis, as I'll come to later. So the function of the TOR protein, besides being the target of the binding site of rapamycin in the cell, work from our lab and a great number of other labs showed that the role of TOR is to control cell growth and thereby cell size. And this is most photogenically shown in these experiments, which were largely confirmation of what was already known, done in Drosophila. And this was in this experiment by Tom Neufeld, University of Minnesota, and then this experiment by Bruce Edgar, now at Utah. And what Tom did here was he selectively inactivated TOR, and this, of course, underscores how highly conserved TOR is. You can work on it in any eukaryotic organism. He selectively inactivated TOR in these 2 GFP-expressing cells, and as you can see, these cells end up being smaller than the surrounding wild-type cells. Bruce did the inverse experiment. Instead of inactivating TOR, he hyperactivated, and he found, again, marking those cells where the change occurs with GFP, and he can see that the TOR hyperactive cells are much larger than the surrounding wild-type cells. In both cases, this is the fat body of the fly. So the obvious conclusion from this is that TOR controls cell growth and thereby cell size. Now, Tom Neufeld took this experiment one step further. He isolated a TOR mutant fly in which every cell of the whole fly is defective for TOR signaling. It's important to point out that this is a so-called hypomorph. In other words, a partial loss of function. This is a mutation in the promoter which reduces TOR activity to about 20% normal levels. That explains why this fly is viable. But although it's viable, as you can see, it's a perfectly proportioned but smaller fly. And then Tom asked the question, why is this fly smaller? Is it smaller because it has fewer cells, or is it smaller because it has a normal number of cells, but now each cell is smaller? And how would you do an experiment to answer this question? Well, what he did was he looked in the wing, because the wing is a plane of cells where you can easily measure the circumference of cells and also count the number of cells. And then from that you can extrapolate to the rest of the animal. And that's what he did, and he found that this fly has more or less the normal number of cells, but each individual cell is slightly smaller, to end up with a smaller but perfectly proportioned fly. So the conclusion from this is that TOR controls not only cell size, but as a result controls size of tissues and ultimately size of the whole animal. To my mind, this is an absolutely astounding experiment or result, because I never thought this would have been possible, because to create a fly like this is a program in 4 dimensions, and here this program is being rewritten in all 4 dimensions by, or at least 3 we can see, 2 we can see here. It's been rewritten in all 4 dimensions by a single mutation in the genome, in this case in the promoter of the TOR gene. You can think of all the metabolic circuits, regulatory circuits, which have to be rewired somehow to create this smaller but perfectly proportioned fly. I never thought that would have been possible. Okay, it's also important to realize that TOR controls growth in many other physiological contexts. For example, in the liver, if you eat or fast, your liver changes in size, and this is due to change in size of liver cells. You don't acquire more cells or lose cells depending on your diet. You know, if you exercise, your muscles get bigger, and again, you don't acquire more muscle cells. Your individual muscle cells get bigger. This has all been shown to be TOR controlled. TOR is actually broken into popular culture, gone mainstream here. This is something you can find on the internet. It's a dietary supplement for bodybuilders. And if you look in the label, it says mTOR and FOXO and PK and all these things, which I'm sure these bodybuilders know exactly what they are. I don't know what's in the flask. I think just leucine, for reasons I'll come to later. But it gets worse than this. I've always wanted mind-blowing results. You can also find this on the internet. Okay, but let's get back to more serious matters. In the central nervous system, there's a very dramatic change not in cell size but cell shape, and TOR also controls this. And someone has actually shown a queen bee is bigger than worker bees because the TOR pathway is upregulating the larvae, which are fed royal jelly, which then induce hypergrowth in a queen bee. So the take-home lesson here is that TOR controls growth very broadly. Broadly in terms of organism and broadly in terms of physiological context. Essentially any case in eukaryotic biology where you see a change in cell size, there's a high likelihood that TOR is somehow involved. So how does TOR actually control cell growth? Well, it does it by controlling a large number of cellular processes, and this is all elucidated over the years from about 1995 to about 2005. It does it by controlling a large number of cellular processes which collectively determine mass accumulation and thereby cell size. And these cellular processes can be subdivided into two groups, the anabolic processes, which TOR activates, and the catabolic processes, which TOR inhibits. So what TOR does is it balances these opposing forces of synthesis and degradation in response, and this is important, in response to nutrients. So in very simple terms, TOR is what makes us accumulate mass and grow when we eat. So how does TOR actually control all these processes? Well, there are effector signaling pathways which emanate from TOR, and these pathways intersect with key proteins involved in all these processes. In some cases we know what those pathways are, but in some cases we do not. But the picture which emerged, and this is where we were in the year 2000, and this is a yeast cell, and please don't get distracted by all the individual proteins in these pathways. I show this just to give you an overview of what I can call the architecture of the TOR signaling network. And we call it a network because what emerged is that TOR is more than a single signaling pathway. It's actually two signaling pathways. And here you can see this. One of the two pathway in yeast, we call this sometimes the TOR2 unique pathway because only TOR2 can signal through here. And this controls organization of the actin cytoskeleton, which in turn controls the secretory pathway, orients the secretory pathway, and thereby determines where new mass is laid down in the cell. So we view this pathway as mediating spatial control of cell growth. The other pathway over here, this, unlike the TOR2 unique pathway, can be mediated by either TOR1 or TOR2 redundantly. They're interchangeable in this branch over here, or this pathway over here. And this then leads to all these processes which lead to actual activation of macromolecular synthesis and mass accumulation. And this is what responds to nutrients. So I think the logic of these two branches, and this then we view as mediating temporal control of cell growth. So the logic of these two branches we think is they integrate spatial and temporal control of cell growth such that the cell knows when and where to grow. And of course these two aspects of growth have to be integrated for growth to occur properly. Before I forget, this is actually we're having two TOR proteins actually helped our analysis in yeast because we were able to identify genetically over about 10 years these two different signaling pathways in yeast based on their differential requirement for the two TOR proteins. Had there been a single TOR in yeast, for example, TOR2, and we knocked that out, we would have lost both pathways simultaneously and never would have seen that there were two pathways. So here we could knock out TOR2, we lost this pathway, but this still was fine because TOR1. We could knock out TOR1, nothing happened, and then we knock out both, and then TOR1, TOR2, we lose both pathways. So having two TORs allowed us to identify genetically the fact that there were two pathways. The next major breakthrough occurred in 2002 with the biochemical identification of the two TOR complexes. And this we did by purifying the two TOR proteins directly out of yeast cells. And we did this because we wanted to understand what were the direct effectors and regulators of TOR, which the genetics was not giving us. We reasoned if we developed a gentle enough purification scheme, we could purify TOR. If it had interacting proteins, we could co-purify them and thereby identify them via this co-purification. Well, we indeed purified these two complexes, which we called TOR complex 1 and TOR complex 2. Once we had them purified in the test tube, then we could sequence the proteins. From that information, we isolate the genes. Once we had the genes, we could knock them out. And we did that. We realized that two complexes corresponded to these two previously identified pathways of TOR signaling. So this is a major breakthrough because it provided a molecular basis for this complexity of TOR signaling. It said TOR is, in fact, two structurally and functionally distinct kinases signaling through two different signaling pathways, and the specificity of the TORs is determined by which complex the two TORs assemble into. And he also explained why either TOR can signal here, because either TOR can assemble into this complex, but only TOR2 can assemble into this complex. Okay, so this is where we were in 2002. We then wanted to know whether the TOR complexes were conserved like TOR itself. So then we went into the genome database, the human genome database, which was not yet complete in those days. And we found a number of proteins which had some identity to the yeast proteins which made up the two complexes. So this then suggested the complexes were conserved. So we, but also David Sabatini, Kazuyo Nozawa, and Kunliang Guan started working on these mammalian orthologs. And we found that the two complexes are conserved, made up of the same proteins, or orthologs of the same protein that make up these two complexes. In yeast, controls many of the same, or acts on many of the same proteins downstream to control many of the same processes involved in cell growth, and also is activated by nutrients in mammals, shown by Joe Averick first. Mammalian TOR responds to nutrients, amino acids in particular. And leucine is a particularly good amino acid among the amino acids, which is probably why there was leucine in that flask for the bodybuilders. So the picture that emerged now by this stage is about 2005, I would say, was that this is a primordial or ancestral signaling network which has been conserved throughout eukaryotic evolution to control this very fundamental process of cell growth. Now, the exception to that statement I just made is this part of the network up here, which is the growth factor signaling pathway, which evolved later. This evolved in multicellularity and then was grafted onto the more primordial TOR network, which already existed in unicellular organisms. And the reason for that is that controlling growth in metazoans is more complex than controlling growth in unicellular cells. And the reason for that is that in a multicellular organism, it's key to control the growth of every cell in the body with every other cell in the body, such that our organs end up being properly proportioned. So you need an input which controls growth over a whole body plan. And this is why the growth factor, why I assume the growth factor pathway evolved, or was grafted onto the TOR pathway. So in humans, we know, or in mammals, or higher eukaryotes, multicellular organisms, we have input via growth factors, which control growth over a whole body plan. We know we need also local nutrients, and now we also know we need local energy in the form of ATP. So these are the three inputs which control the TOR network: growth factors, nutrients, and energy. And once they're present, then the TOR signaling network is activated, macromolecular synthesis ensues, and growth follows that. Interestingly, the entrepreneurs were not far behind. You could also find this on the market shortly after discovery of the TOR complexes. Now, another fascinating aspect of the TOR signaling network is the unusually large number of diseases to which it's been genetically linked. And interestingly, what all these diseases have in common is they're characterized by inappropriate or ectopic cell growth, which also underscores the central role of TOR in controlling cell growth. And this can be either malignant forms of cell growth or benign forms of cell growth, but in all cases, inappropriate cell growth. It's been calculated TOR is upregulated and contributes to tumorigenicity in 70% of all cancers. And this is something I'll get to when I start talking about current work from the lab. More recently, TOR has been implicated in another set of disorders, the so-called metabolic disorders. We know that chronically high circulating levels of nutrients can upregulate TOR even in the absence of growth factors, and this can lead to adipogenesis and obesity, which then, as you know, can lead to type 2 diabetes. But there's a more direct link to diabetes, this famous negative feedback loop in the TOR network, where mTOR phosphorylates and inhibits IRS, the insulin signaling pathway, to to confer insulin insensitivity. And of course, this is a hallmark of type 2 diabetes. So in this case, some have actually proposed rapamycin could be used as an anti-diabetic drug, because if you inhibit TOR, you'd inhibit the negative feedback, and then you'd restore signaling through the insulin pathway, and then restore insulin sensitivity. I don't think anybody's actually developing rapamycin as anti-diabetic because there are a number of other complications, but something that gives credence to this notion is the fact that the world's most commonly prescribed anti-diabetic, metformin, acts at least in part by activating AMPK, which then inhibits mTOR. So there's some support of this notion. Okay, another fascinating aspect of physiology of TOR is that it controls lifespan. This was first shown in worms, then in yeast, then in flies. In this landmark paper by Rich Miller in 2009, it was the first physiological demonstration of, first pharmacological demonstration of lifespan extension in mammals. And this is, and what he did was he gave rapamycin to mice and found he could extend lifespan in mice. This created a great deal of excitement in the aging field because it's been known since 1935 that caloric restriction can extend lifespan. But the molecular basis of that was not known. And now we know TOR is a nutrient sensor, and the effect of rapamycin is to mimic caloric restriction. It tricks the cell into thinking there's less food there, less calories than are really there. And so it provides now a model or a mechanism for how caloric restriction extends lifespan. If you eat less, TOR is less active. This alters metabolism in a way which the net effect is beneficial to living longer. Okay, so this is where we were about 10 years ago. And about 10 years ago, there was what I thought was a really astounding discovery made by another fly geneticist, Pierre Leopold, who was at Nice, now in Paris. He was looking for flies which had a growth defect, much like the fly which Tom Neufeld had found earlier, which I showed you earlier. But the mutant that Pierre isolated was defective in an amino acid transporter, which was specific to the one tissue, the fat body. So as a result, amino acids didn't get into the cells of the fat body. As a result, all the cells of the fat body had inactive or less active TOR. As a result, the cells of the fat body were smaller than the fat body. This one tissue was smaller. Completely expected based on everything we knew at that time about TOR signaling. Now what he discovered, which is what I thought was so astounding, was that not only was the fat body smaller, but all other tissues which have their own amino acid transporters, not the same one that's found in the fat body, all those tissues, and this is all different tissues, all these different tissues were also correspondingly smaller. So this told us that there was a TOR-dependent signaling between tissues to control growth over a whole body plan. That somehow all the tissues can readjust their growth to be the proper size for whatever the rate-limiting growth is for a particular, whatever tissue is rate-limiting for growth. So this opened up to my mind a completely new vista, a new area of growth regulation. What is this non-cell autonomous control of cell growth between tissues to arrive at that controls growth over a whole body plan? And we then started studying that, but not in flies, in mice. And what we had to do now was understand the role of TOR in every tissue, but not only the role of TOR, the role of TORC1 and TORC2 separately in every tissue. So we made mice which were flocked for Raptor or Richter to knock these two proteins out. Raptor is an essential and unique specific subunit of TORC1, and Richter is an essential and specific subunit of TORC2. So we could knock out TORC1 and TORC2 individually, and then we could start looking at different tissues. So since TOR is present in all tissues, we had to decide what tissue to work with, and we decided to work on the so-called metabolic tissues. And the reason for that is that these are the tissues which are most nutrient sensitive, most insulin sensitive, and the tissues which control energy homeostasis in the body. And these are precisely the same three inputs which control TOR. So we decided that TOR would have a particularly interesting or important role in these tissues. So we've done that, and we've also sent these mice out to anybody who's asked for them with no strings attached. with the idea that it's like a Manhattan Project. In the end, we'll try to collate all this data from all these different labs to see how TOR in different tissues controls whole body, contributes to whole body growth. So just let me give you just a quick taste of what we found when we knocked out TOR in adipose tissue. This is what happens when we knocked out TOR complex 1, specifically in adipose tissue. The mice are resistant to diet-induced obesity. Metabolically, they're even healthier than wild-type mice. They have low levels of insulin, low levels of cholesterol. They all have full-body metabolic changes, again, supporting what Pierre Leopold saw that there are non-cell autonomous roles of TOR in signaling to other tissues to control, in this case, whole-body metabolism. This also, given that this mouse is even healthier than the wild-type mouse, harkens back to the fact that rapamycin extends lifespan. So that experiment that Rich Miller did, he gave rapamycin to mice, which inhibits TOR in all tissues. But the question remains, does inhibiting rapamycin in a particular tissue give this beneficial effect? And this then suggests that TOR signaling in adipose tissue plays a particularly important role in controlling lifespan. We haven't done the experiment to see if this mouse lives longer, because unfortunately the mice live longer than PhD students in the lab, and no one's been altruistic enough to set up the experiment for the following student. So that's what we found, TOR complex 1 in adipose tissue. In knocking out TOR2 in adipose tissue, we were very surprised to see the mice even get bigger. Again, indicating their non-cell autonomous functions of TOR in whole body growth. And this then led us to discover a mechanism of insulin in IGF, insulin-like growth factor homeostasis, in which TOR complex 2 in adipose tissue somehow negatively signals to the liver and the pancreas to inhibit IGF production from the liver and insulin production from beta cells. And the logic behind this is that TORC2 itself is controlled by IGF and insulin. So this, if you have too much insulin in IGF, this inactivates TORC2, which reduces their synthesis. So this is a mechanism of maintaining homeostasis. But in the case of the mice, if we knock out TORC2, we lose this homeostasis. These mice have very high levels of IGF and insulin, which leads to these mice growing bigger. This is increasing lean mass. Their muscle mass is more. So this is where we were, again, about up till less than 10 years ago. We've continued to knock out TOR in many different tissues, looking at many different physical contexts. And while we're trying to understand the physiology on a whole body, in a whole body context, we've also gone more molecular, more atomic. We worked out the structure of TOR complex 1. We've now gotten down to less than 5.9 angstroms, but this is what we published in 2016. This took us 10 years to figure out this structure. TOR is this is TOR complex 1, the human TOR complex 1. It is a dimer of heterotrimers, 2 catalytic sites, a catalytic cleft here and here. The gray and the blue are the 2 TOR dimers. This is Raptor in the green. This also allowed us to elucidate how rapamycin FKBP works, because we also have the structure of FKBP rapamycin. So the red is FKBP. Let me go back and forth. You add rapamycin, FKBP, using rapamycin as the glue, binds the lip of the catalytic cleft, and there sterically hinders substrates gaining access to the substrate. So this is how rapamycin works. This is also shown by the Pavlich lab in New York, who made the crystal structure of the catalytic domain of just the catalytic domain of TOR complex 1. So as I said, it took us 10 years to get this structure, and the reason is this is a very large structure. We tried many approaches before we finally got it with cryo-EM. We actually did crystal structure of Raptor, and then cryo-EM of the whole structure, and could then dock the crystal structure of Raptor, which we have down to 3 angstroms, into this cryo-EM determined structure. So this is the whole TORC1. But the reason it took us so long, the reason we needed cryo-EM, is that this is a very large structure. But you can appreciate that from here. So to give you a feel for the size of this, I'll stand it up and then give you a reference molecule, which is the ribosome. So TOR complex 1 is about as big as the ribosome. And what should immediately strike you, of course, is this is a very dense structure, and this is a very open structure. And I think this reflects our limited knowledge on TOR complex 1, on the regulation of TOR complex 1, what the proteins are that bind TOR 1 to activate it, such as the RAGs and the REBs. And this is all work that is being done at this moment, not only by our lab, but a number of other labs to understand how TORC is regulated at the atomic level. Earlier this year, we also finally got the structure of TOR complex 2. Still, we're trying to improve the resolution. This is about 7 angstroms. But this revealed something which we had bothered us for a long time. And that, as we knew, also something we knew since 2004, that TOR complex 2 is rapamycin insensitive. So why could rapamycin inhibit TOR in TORC1 but not inhibit TOR in TORC2? And we got the structures. It was evident that the reason for that is that whereas FKBP can bind the lip of the catalytic complex in TORC1, the Richter submutant in TORC2 masks that binding site which originally identified genetically in yeast, such that FKBP rapamycin cannot bind TOR complex 2, and that explains why TOR complex 2 is rapamycin insensitive. Okay, I've come to the end of my introduction. I'll give you an overview of a little bit of the history, the physiology, the function of some structural biology on TOR complex, on TOR across species from yeast all the way to humans, and I've summarized almost 30 years of TOR research. Let me tell you something that's going on in the lab now, and this has to do with, I'll give you 1, 2, and possibly 3 stories, depending on the time I have. They can all be bundled together under this general title of mTOR in liver cancer, so the lab has gone more and more into the role of TOR in disease. And the first problem I will talk about is the role of mechanisms of evasive resistance to targeted cancer therapies. So this was a dramatic change in research for my lab because it's a translational research project. Actually, I'm very happy I did this because I think we're at a point in time where there are really unique opportunities for basic scientists to go into translational research. Their technology has gotten to a point where we can actually do interesting experiments in translational research. But how do you define translational research? I think everybody you ask will have a different definition of translational research. The best definition I've ever heard, the one that works the best for me, is that it is research in which a basic scientist comes into physical contact with the patient. So here's the evidence that this is a translational research project. Of course, I'm doing this, and this is important, via a clinician. Now, the real reason I show you this slide is so you can see this down here. This is a long coaxial needle that the clinician is using to perform a biopsy procedure on this patient. He's, in this case, doing a biopsy procedure on the liver of this patient. And this biopsies are at the core, key aspect of this project I'm going to talk about. I should say this is not a completed story. It's a progress report. It's something we started about 4 years ago, and we're continuing to work on it. I just thought I'd give you a taste of this project with this progress report, but don't expect any answers. So here's the actual problem. These are 5-year survival rates of patients with different kinds of cancers. This is what they were many years ago. This is also more recently, but also quite some time ago. And as you can see, there have been improvements, but these improvements have been rather mild. And I think immunotherapy will increase these numbers here, but not that significantly. So this has been a disappointment. Why are cancer rates not being why is cancer not being cured? And this disappointment is compounded by the fact that over the last 40 years, the signaling community, many of the people in this room, have been elucidating the so-called oncogenic signaling pathways, which in turn has led to the production of this huge arsenal of drugs which are targeted therapies which inhibit, in this case, these are all kinase inhibitors. So there have been about roughly 30 over the last 30 years, and I hope you'll see your drug is in here. I can't, I don't know. So this is a big disappointment. Why are these cancer rates, cure rates, not going up despite all these new drugs which are going on the market? And the reason is that whereas these drugs work, they don't work very well. And this is illustrated here. This is in the case of this one drug, Avastin, for colorectal cancer. With drug, there is a benefit. It's a very small benefit. In this case, there is a 6-month benefit of progression-free survival. This is one drug. Here's another one. This is Definitive for lung cancer. In this case, there's about a 6-month benefit. You can go through this with essentially every drug on that slide I showed you earlier. So why aren't these drugs working as well as we hoped? And there are two reasons. One is that tumors are intrinsically resistant. In other words, they don't target the defect. And this sometimes stems from the fact that cancer therapy is a one-size-fits-all therapy, but not all tumors are the same. So they're just, we need to personalize medicine more to understand which drugs to give to which patients. The second reason is that the patient responds to the drug, but then develops resistance. And this is referred to as evasive, adaptive, or acquired resistance. And this is actually what happens most of the time. So let me give you a little very simple cartoon to illustrate this evasive resistance. We know different pathways control cell proliferation. When one of them gets ectopically activated by a mutation, say activation of an oncogene, this leads to inappropriate growth or a tumor. What the targeted therapies do, of course, is target these pathways to inhibit them. If the tumor is responsive, there'll be a regression. But what invariably happens, there is a new mutation which appears either after therapy or was present at some low level in the tumor itself, which activates another pathway, and in response to continued selection by the drug, these resistant cancer cells will grow out, and you'll have a relapse and tumor progression. Now, the problem is that we don't know so there's a compensatory pathway which is activated to circumvent the drug block, but the problem is we do not know what these compensatory pathways are. We don't know how they're activated, nor do we know how they actually compensate once they are activated. To my mind, this is one of the major challenges in cancer research today. So just to give some actual pathways to this cartoon-like presentation, here's what's found in preclinical research. Also underscores how little is known about this. It's been found that in melanoma cells, skin cancer cells, those cells which are resistant to common drugs used to treat melanoma, the resistance to these drugs correlates with hyperphosphorylation of S6, which is a downstream effector of mTOR. Similarly, breast cancer, resistance to PI3 kinase inhibitors also correlates with hyperphosphorylation of S6. So it's important to know these things because it shows that S6, phospho-S6, can actually be a biomarker to inform the clinician not to treat with these drugs because the tumor will be resistant. It also tells that a more effective therapy might be a co-therapy with a TOR inhibitor in combination with this or this. So it's important to understand what these compensatory pathways are and how this information could be exploited for better therapy. I should say it's not known also in this case what activates TOR in the melanoma or breast cancer cells, nor is it known how TOR, once activated, it can compensate for this pathway or this pathway. It does seem that pictures emerge that TOR seems to be a central compensatory pathway, and this might reflect its very broad role in cellular physiology. It can compensate for many different pathways which are blocked by different drugs. And this is one of the reasons we were sucked into this problem. So we wanted to address this problem. We decided to focus on the liver. And we focused on the liver for a couple reasons. One is there's a medical need. There's no cure for advanced liver cancer, hepatocellular carcinoma in particular. And the second reason is that the liver is a tissue which you can serially biopsy. So you can do a longitudinal study, and the biopsies you get are large enough so you can do the kind of biochemical analyses that we want to do. So we considered all tissues, and we realized that after we checked all the boxes, liver was the only tissue we could work with, only tissue which satisfied our needs. And just to show you that liver has a problem with targeted therapies just like all the other tissues or drugs. In the case of HCC, the target therapy of choice is sorafenib. Again, like all the other drugs I showed you, it works but not very well. In this case, there's about a 3-month benefit to sorafenib treatment for patients with advanced HCC. So resistance to sorafenib is also a problem in the case of HCC. So what are we doing? The patient comes into the clinic. We perform a biopsy procedure, like I showed in that slide, with these long coaxial needles. We take a biopsy not only of the tumor, but non-tumor tissue from the same patient, so we have a control. And then we immediately snap freeze the tissue to preserve the in-tumor properties of that tissue. I have to tell you, as an aside, we originally were going to work with resected tissue until I learned, to my horror, in casual conversation with the surgeon, that the way they perform their tumor resections is by clamping off the blood supply to the tumor. And what this does is, of course, would make the tumor hypoxic and hypoglycemic, which would completely scramble all the signaling pathways, the dynamic signaling pathways in the tumor. And that's, of course, ultimately what we want to look at, the signaling pathways in these tumors. So I had no idea what the surgeon did, and the surgeon had no idea what was important to me. It was only this casual conversation that I realized that we cannot use resected tissue. And I say this is a lesson to anybody who wants to start doing translational research. Talk to the doctor, make sure you know what he's doing. Go to the operating room, see what they do, because it makes a big difference. And I think this might be responsible for why biomarkers often don't work, because they're in this case, what we found would have been a biomarker for the intervention, not for the disease. Okay, so we quickly changed what we did to doing biopsies where it's minimally invasive and you can immediately snap freeze the tissue. It comes out. Our biopsy is about 10 milligrams, from which we get about a milligram of protein to work with. After we do this initial biopsy, then therapy starts, in this case sorafenib. Then we continue to monitor the patients, again taking biopsies at regular intervals, these paired biopsies. And once we have all these paired biopsies, we then apply a whole battery of omics analyses, exome, transcriptome, proteome, and most importantly, phosphoproteome, because this gives us a snapshot of all the signaling pathways, which is what we're interested in. But the first thing we do, and this is something I completely underestimated the importance of as a basic scientist, was we show the biopsies to our pathologist, who gives us the green light that, first of all, the pathologist doing the procedure didn't miss the tumor. This is actually tumor tissue, and also gives us indication on how clean these biopsies are, how much infiltrating immune cells there are, for example, which could confound our analysis. Okay, so once we, so this is how it works. This is the workflow. It starts with the patient biopsies, all these omics analysis. This generates a huge amount of data, which we then try to integrate. And this is the bottleneck so far in this whole project. There's no off-the-shelf software which allows integration of so many different kinds of omics analysis. It's something that we have to do ourselves with working with computational biologists. But once we are able to do that, we can generate hypotheses which we can then test going back to mouse model or cellular systems, or if the patients are still alive, which usually they're not, we can alter the therapy in the patients. Okay, so most of this is rather conventional, genomics, transcriptomics. What is not conventional, I don't think anybody had ever reported when we started this, a phosphoproteome or a proteome on a tumor biopsy. So let me just focus on this proteomic, phosphoproteomic part in this for a couple minutes. What's important here is that we do quantitative proteomics. Because what we're doing is comparing tumor tissue to non-tumor tissue from one patient, from one time point, but also from different time points from that one patient, also from different time points of many different patients. So, and this is against a background of great heterogeneity in gender, age, diet, and disease. So this is a very risky project, trying to compare all this data from such a diverse from such heterogeneity. And this is why quantitative proteomics is important. And the quantitative proteomics we do is SuperSciLac, a method developed by Heitmann, where we can spike in a standard. We don't have to in any way manipulate the tissue once it comes out of the tissue, once that comes out of the patient. We developed the method to do this, and the amount of protein we get, I should say, is at the limits of what you can work with to do proteomics and phosphoproteomics. We were able to do this. We could get a phosphoproteome from a test biopsy from many signaling components, and we could even measure, detect, and quantify signaling phosphorylation changes in the signaling pathways which are commonly activated in HCC. This is the RAS MAP kinase pathway, the PI3 kinase mTOR pathway, and the Wnt pathway. These are the common pathways which are activated in HCC. So the method was working, and we could now start working through our cohort of patients, which we'd started building. Of course, we had to get ethical approval to do all this. And here's the first patient. We started with a very simple patient. The patient, there was paired biopsies, control and tumor, before sorafenib treatment, and then a second one on sorafenib treatment. This patient seems to have been intrinsically resistant to the drug, so sorafenib treatment was stopped. And this intrinsic resistance is illustrated here by alpha-fetoprotein levels, which increase, which is a marker for the tumor, and of course, the patient died shortly thereafter. So we had four biopsies to work with, control and 2 tumor. This again illustrates how this patient seems to have been intrinsically resistant to the drug. The drug had no effect at all in slowing down the tumor. And this is another very nice aspect of the study, is we have full clinical records of all these patients. Okay, these are the biopsies which we showed to the pathologist. He said they were good and we could work with them. And I like to show, so this is the amount of protein which we could, the protein yield from each one of these 4 biopsies, control and tumor. And I like to illustrate this because in one case we got only 100 micrograms of protein to work with. And this was really, we almost gave up at this point. We thought this was too little protein to do a proteome and a phosphoproteome. But this also illustrates the vagaries of doing translational research. We couldn't go back to the lab and culture more cells or put up a new mouse cross. We had to forge ahead with the material we had, because this is of course non-renewable material. This psychologically was a big change for me. How it really the postdocs who did this require a lot of courage and nerves of steel. Okay, so this is what we found then from that one patient. This is now comparing tumor before sorafenib, tumor after sorafenib. And what we learned was that in these are the various signaling pathways. Blue are phosphorylation sites which are downregulated or lost upon sorafenib treatment. And this tells us that the sorafenib actually worked as advertised in this patient, which was intrinsically resistant to the drug. It hit its target and inhibited. Downstream effectors were hypophosphorylated, just like you'd expect. To my knowledge, this might be the first demonstration that sorafenib actually works in a patient the way it's supposed to. But we also learned that if we look further downstream in the pathway, effectors further downstream, phosphorylation of these is unchanged or even increased. So there's something else coming in downstream here, which is maintaining these further downstream effectors phosphorylated and activated in the presence of this upstream block. We don't know what that other thing is, and this is something we have to now figure out. We're working through our cohort, and once we have all the data, then we'll be able to crunch all the data together and get some meaningful information to what might actually be going on. But this just gives you a taste of what we're doing. Every patient is different too. This is another patient. In this case, we did 3 biopsy procedures. And in this case, there was a very large tumor, so we could actually biopsy the same tumor more than once. So this will give us an indication of the effect of sorafenib on tumor heterogeneity. Every patient gives us new kinds of information. This is another patient. This one is our record. Most number of biopsy procedures, 4 biopsy procedures. This patient had metastases. So we not only biopsied what we think is the primary tumor, but peripheral tumors as well in the liver. And once we do our omics and genomic sequencing of these of these biopsies, we'll know which was the primary, which were the metastases. But again, we'll be able to learn the effect of sorafenib on not only primary but metastases. And this is what we're working through now. I think the last patient of our, so far we're going through a 10-patient cohort. Our last patient should be finished by early next month, and then we'll be able to get some meaningful information out. So this is the whole project. I told you about, I just focused on phosphorylation, phosphoproteome and phosphorylation levels, but there are these other dimensions too, RNA, DNA, which we're also looking at, which will generate these hypotheses about what confers evasive or intrinsic resistance, and also will hopefully lead to personalized interventions. This is work not only from my lab, but it's the work of a ERC Synergy grant, which we obtained. It's a small network where, including Gerhard Christofori, who is a tumor biologist, Nico Barenwinkel and Jörg Stelling, who are both computational biologists, who are extremely important for this project. Okay, so that's the sort of update on that project. If I come back in a few months, I think we'll have a lot more information there. So here's the second story. It's an extension, perhaps, of what Tony Hunter talked to you about earlier on. He said he mentioned this, but only lightly. So let me go into this in a little more detail. In addition to working with those patients, we also generated a mouse model for HCC to complement our work with patients. I was, of course, nervous that we wouldn't get the patients or nothing would work with patients. It's much more difficult working with patient material. So we created this mouse model to help us with the patient work. And this stems, and what we did here was we made a mouse model for HCC. We did an mTOR-driven mouse model for HCC. And this was motivated by the fact it's been known, as I told you before, that mTOR is a major driver in cancer. But the effector pathways by which mTOR causes cancer are poorly characterized. So this is what we wanted to understand. So this is the mouse model. Again, we're back to the mTOR signaling network. We made this mouse model by deleting 2 tumor suppressors in this network and doing this specifically in the liver. And the 2 tumor suppressors we deleted were TSC here and P10 over here. So when we delete TSC, we hyperactivate mTORC1, and we delete P10, we further activate mTORC1, but now hyperactivate mTORC2. So and we do this specifically in the liver, as I said. So we have mTOR network hyperactivated in the liver. So what happens here when we do this? And we call these mice LDKO. You'll see this in the slide. So this liver-specific double knockout. So the first thing we see was that it's hepatomegaly, or enlarged liver, and this is due, of course, to the growth-promoting role of TOR. TOR is activated in the whole liver. All the hepatocytes in this liver get larger, and the liver gets larger. We already see this at 4 weeks of age. The knockout occurs at about birth. By about 6 to 8 weeks, we start seeing steatosis, or fatty liver, which then progress in the liver, as you see, continues to enlarge. Again, it's continued TOR signaling. By about 10 weeks, we start seeing inflammation in the liver. So the fatty liver progresses to inflammation, and this then progresses to about 16 weeks to very mild hepatocellular carcinoma, and then by 20 weeks we have very, very severe HCC. And the mice died about 20 weeks of age. We showed this to our pathologist. He said this is bona fide HCC. So it seems to be a good system to study HCC. There are 2 points I'd like to make about this system. The first is very faithful. We've looked at hundreds of mice, and we've yet to see one which does not develop HCC and with this time course. So we can add something here and assume that if it inhibits, there would have been HCC. The other is it recapitulates what's seen in the clinic, where it's known that fatty liver is a risk factor for HCC. And this is exactly what we see in this mouse model. Now, so we were very confident this was a good system, but we want to do one control experiment. Is it indeed an mTOR-driven mouse model for HCC? And to answer that question, we treated the mice at 11 weeks with rapamycin, and And then at 20 weeks, we examined the liver. And what we found that if we rapamycin treated 11 weeks, the liver looks a little fatty but relatively healthy. And as usual, if we didn't treat, the mice displayed this very severe HCC. So everything checks out. The next step was then to start studying these tumors. And the questions we ask now are, what's giving rise to these individual tumors? Do they have common events? What are these events that give rise to these tumors? Are there common events? Are they all independent events? And to answer this question, we dissected out the individual tumors, and again, through all our omics analyses at them. And this is what we found. So we looked at 12 tumors, 3 each from 4 different mice. And as a control, we compared these to 6 pooled wild-type livers. And these are then the amount of proteins. I'm going to focus on the proteome now, although we did all these other kind of omics analyses. And we looked, so these are 12 tumors. We could detect and measure about 5,000 proteins per tumor, about a third of which were upregulated and about a fourth of which were downregulated in the tumor controlled to the liver. If we look at these numbers more carefully, we could actually detect and measure 8,000 proteins across all 12 tumors, but if we limited our analysis to those proteins which are detected in at least 10 tumors, we come down to about 3,000 proteins. And of these 3,000, 400 were upregulated and 262 were downregulated. So that we then further focused our analysis on these proteins. And Shravant, the postdoc who did this, then to narrow the analysis even further, looked specifically at kinases and phosphatases that are up or downregulated, And one kinase in particular caught his eye, NME1 and 2, and one putative phosphatase, LHPP. And these caught his eye because these as Tony probably told you, this is a histidine kinase, and this is a putative phosphohistidine phosphatase. So this suggests that histidine phosphorylation is different in these tumors, is up. There's more histidine phosphorylation, and this might be responsible for the tumor. Now, when I started this, when I made this observation, I knew very little about histidine phosphorylation. You probably know more than I knew based on what Tony told you, but let me just give you a quick update, refresh your memory since I guess Sunday. So as he said, probably said, histidine phosphorylation is a hidden phosphoproteome. For reasons I'll tell you in a moment. We all know these 3 amino acids can be phosphorylated. Several others can as well, including histidine. However, unlike threonine and tyrosine phosphorylation, which is a PO bond, histidine phosphorylation is this is stable. Histidine phosphorylation is extremely labile. And this is due to the nature of the PN bond versus the PO bond, which is formed during histidine phosphorylation. There are also 2 isomers of histidine phosphorylation, depending on which nitrogen in the imidazole ring of histidine is phosphorylated, the 1 position or 3 position. I'm going through this quickly because I'm sure Tony told you about it. NME1 and 2 are the known kinases. There are 2 known phosphatases, PHPT1 and PGAM5. And then there's LHPP, which is a suspected but not shown phosphatase. One last aspect of histidine phosphorylation is the mechanism of phosphorylation, which is also interesting. NME1 and NME2 are homologous proteins. They form homo or hetero oligomers, and each one of these oligomers phosphorylates itself on a specific histidine in the 1 position, and then it transfers that histidine to the 1 or the 3 position in substrates. Back to our data. So this is the 12 tumors. NME1 and NME2 was up in all 12 tumors. LHPP was down in almost all 12 tumors. The 2 known phosphatases were essentially unchanged. So there's something special about LHPP. We confirmed by blotting what we'd seen in mass spec. The LHPP is gone in the tumors compared to control. NME1 and 2 are up in all the tumors compared to control. We want to know where these changes were specific to the tumor versus non-tumor tissue of the same liver. So here we dissected apart tumor tissue and non-tumor tissue. It's also shown by immunohistochemistry on this side. If we look in the tumor, this is LHPP. We lose LHPP specifically in the tumor, non-tumor tissue from the same liver still expresses LHBP. NME1, on the other hand, is up throughout the liver and the tumor. So NME expression follows hyperactive TOR expression. LHBP does not. So it seems like loss of LHBP in a background of high TOR signaling where NME is high might be what's driving tumorigenicity. So we think NME is controlled by TOR, but LHBP is not. So this is where we were a few years ago. We thought histidine phosphorylation was important for tumorigenicity, but how are we going to study it? Remember, it's a hidden phosphoproteome. All the biochemical manipulations you do to study it destroy it due to its lability, heat and acid sensitivity. And it was at a meeting where I ran into Tony, who told me about his monoclonal antibodies, which I'm sure he just told you about. And they were at that time unpublished, but precisely the reagents we need at that moment to start looking at our tumors. This was like manna from heaven for us. So we immediately started a collaboration and immediately probed our tumors. And this is probing our tumor versus control with a monoclonal against 3-phosphohis. And you can see there are high levels of 3-phosphohis in the tumor compared to the control. Probed with 1-phosphohis. There's much less 1-phosphohis in the cell. This is in NME itself, the autophosphorylated form of NME. So there's either much less 1-phosphohis or 1-phosphohis is even less stable than 3-phosphohis. So this confirms histidine phosphorylation is up. What happens if we restore LHBP in cells which have lost it? So this is a cell line we made from our tumors. We reconstitute LHBP by introducing it on a virus. So we restore LHBP expression in these cells which had lost it. And when we do that, we also lose these high levels of histidine phosphorylation. So this in vitro work, which I have not showing, confirmed that LHBP is a phosphatase. What are the functional consequences of that, of restoring LHBP? In vitro, if we add back LHBP, we can't get these hepatospheres, which CB1 cells, these cells from our tumors can form. This is in vitro model for tumor formation, but more importantly, we can also do it in vivo. We injected a virus with high liver tropism into the mice, and this is a virus which expresses LHBP, so we can maintain LHBP expression in the liver of our double knockout, our LDKO mouse. And we inject the control. As usual, the mice come down with this severe HCC. If we provide LHBP in a manner which can be turned off, we find there are much fewer and much smaller tumors. So LHBP seems to be a tumor suppressor in mice. We then also went to these biopsies from the human patients, which I talked to you about earlier. Again, we saw in humans what we saw in mice. LHBP has gone in the tumor versus non-tumor tissue, and NME 1 and 2 is up in these tumors compared to non-tumor tissue. And this is shown by immunohistochemistry here. Histidine phosphorylation is also up in the tumor versus the non-tumor. And the clinical correlates of this, we looked at expression data from the Cancer Genome Atlas. Low LHBP expression correlates with low survival. And finally, we looked in cancer genome databases. We found a number of mutations in the LHBP gene, many of which are predicted to be loss of function mutations. So this suggests that LHBP is a new tumor suppressor, both in mice and in humans. So what we think is going on, we think TORC2, and the reason for this I didn't go into, but we think it might be TORC2, which is controlling NME expression, which is then leading to histidine phosphorylation. And this is kept in check by these 3 phosphatases. What we find in our mice is that TOR2 is hyperactivated, leading to higher levels of NME, which is leading to higher levels of histidine phosphorylation, which in the absence of LHBP, without LHBP to check these higher levels of phosphorylation, this leads to tumor progression. We're of course now trying to understand what these proteins are, which when hyperphosphorylated on histidine residue can give rise to tumor. We're purifying these histidine phosphorylated proteins, and then going back to our mouse model to find out what they might be doing. Of course, we're doing this in ongoing collaboration with Tony Hunter. Okay, I have another very brief story. I can tell a third one. Should I keep going? Where's the chair? Oh, but I can do it in less than 10 minutes, I think. Okay. So this is the third and last story I want to tell you about how TORC2 promotes HCC via controlling lipid synthesis pathways. So the story I just told you on LHBP was comparing 20-week tumors to 20-week controlled liver. But what we also want to compare were these, do this longitudinal study of comparing these livers over time to see what the early events are that give rise to these tumors. And this, again, we did many omics analysis, but in addition we did metabolomics and lipidomics in this case. And this is what we found. We found all the enzymes of the fatty acid biosynthetic pathway were upregulated already at 4 weeks of age and continue to be upregulated through into the tumors. And then, of course, the fatty acids are then used to make lipids of many different kinds, but the pathway, the specific lipogenic pathways leading to cardiolipin and glucosylceramide were upregulated. So the many possible lipogenic pathways, it was these pathways in particular which were upregulated. So there's something special about these particular lipids, cardiolipin and glucosylceramide. And these pathways, some of these enzymes are again already upregulated at a very early age of these mice. So so on this side, of course, it's glycerol phospholipids, and on this side, glucosylceramide is a sphingolipid. So two very different kinds of lipids are being affected. We want to see if there's a causal relationship between upregulation of these lipid pathways and tumorigenicity. So we did a similar experiment I described before, virally introduced a knockdown into these of fatty acid synthase or glucosylceramide synthase to see if this would affect tumor development. And in fact, it did. These mice were protected against HCC if we knocked down these pathways, which told us that mTOR is driving cancer via these fatty acid and lipogenic pathways. The question remained, which TOR complex? Is it mTORC1, mTORC2? Because there's been some debate in the field whether it's mTORC1 or mTORC2 which control lipogenesis. As one of the many anabolic pathways which TOR controls. It could be that they're just context-dependent effects here. We don't know. But we did some pharmacological studies which suggested it's TOR complex 2. We wanted to confirm that by now making a triple mutant mouse in which we not only have TSC and P10 knocked out, but we also have now Richter knocked out. So we're knocking out TORC2. And when we do this, so here are the double knockout mouse at 20 weeks, the triple knockout mouse at 20 weeks. It brings, when we knock out TOR complex 2, all the lipid levels come back down to more or less normal levels. We lose also tumorigenicity when we make the triple knockout, and this Kaplan-Meier plot, when we make the triple knockout, the mice live longer than the mice which are just the double knockout. So it looks like TOR is controlling fatty acid lipid synthesis via TORC2. We think this, at least for the fatty acids, is at the transcriptional level through transcription factor SRABP1. It's also controlling enzymes and lipogenic pathways. In some cases, it's transcriptional. In some cases, it's post-transcriptional. For cardiolipin, it seems to be post-transcriptional. For glucosylceramides, it seems to be transcriptional. We're trying to understand why the lipids are important for tumors. We think cardiolipin is a bit of a special lipid because it's found in the inner membrane of the mitochondria where it stabilizes the electron transport complexes. It also lubricates the ATPase to enhance oxidative phosphorylation. So we think it upregulates cardiolipin to enable higher production of ATP, which then would meet the increased energy needs of the tumor. So we think that's what's going on with cardiolipin. We're investigating that now. We don't know what glucosylceramide is for. Lipids can be building blocks or signaling molecules. We have no idea. Glucosylceramides, there are actually hundreds of them, and they're very poorly characterized, and we have no idea what it's being used for. But that's also work for future work. Okay, I've come to the end now. Let me give credit to the people who did the work. The earlier part on the histidine phosphorylation was Shravan, my postdoc, Shravan Hindupur, in collaboration with Tony and Steve Foos in his lab. The work on the evasive drug resistance was the work, another very good postdoc, Eva Datzert, in collaboration with my long-term technician, Marco Colombi, and another technician, Coralie Etter. The structural work, which I also mentioned, was in collaboration with Nenad Ban at DTH and Tim Meyer, a colleague at the Biotentrum. And the lipid part was the work of a student who's just left, Yakir Guri, and also a very good student, and in collaboration with Howard Rietzman at the University of Geneva. And thank you for your attention.